And today I will be talking about some of the work that we have done with in vitro communities of nasal bacteria, and I'll get into more details of it. But before I get into the presentation, I wanted to thank the people in my lab that did most of the work that I will present today. Vaishnavi, Sandra, Sam, and Jawad were graduate students or are graduate students in the lab, and Helen was a former postdoc in the lab. There were also several undergraduate students involved in the work, as listed here, and collaborators that helped us get us started and spend more time with these communities. The overall context of what we will be talking about is microbiota-based therapy. And this is based on the now common knowledge that our associated microbes are important for our health. These could be modulation of the immune system or protection against pathogens as examples. We also know from several examples that disruption of human-associated microbiota can lead to diseases, with examples in eczema and colitis, but there are more examples as well. And that brings up the opportunity that we can potentially restructure and change the microbiota to either prevent diseases or treat them after they have started. This is also important because our common way of changing microbes or affecting them, which has been antibiotics, has started not to work so well anymore because of the rise of antibiotic resistance. So if we find alternatives to affect our microbes without needing antibiotics, that would be helpful. I'll do a quick segue and sort of repose this question that whether or not intervention would be helpful. There is an ongoing debate whether the microbiome state in disease is actually the cause of the disease or not. And there are arguments on both sides, some examples that it seems pretty clear that the cause are the microbes, and several examples that it seems like there is an alternative microbiome state during the disease, but that might be just the response to the disease rather than causing the actual disease. I've put one recent example here. So we know that disease can happen and microbiota exists, but which one is causing which may not be clear in all contexts. But there are these clear examples that we know that the causal agents of disease are linked to microbiota dysbiosis. And those will be the ones that we will talk for the rest of today. So the examples like C. difficile infection or Staph aureus suppression by commensals. So these are known examples. The causes have been studied. So we won't get into the debate of whether the microbiome state and restructuring it is important or not, because we assume that our focus are on cases that it will be important. Back to our microbiota-based therapy. So even though the idea is pretty simple, so say, okay, microbes are affecting us, we change them to affect our health, the actual implementation is not straightforward for several reasons. Most importantly, the microbiota are often complex ecosystems, and it's not easy to change them to any desired state. So there are a multitude of species present. They are engaged in many interactions, either among microbes or with their hosts, and many of those interactions are not well understood yet. And they often have complex and heterogeneous temporal and spatial variations that we don't fully understand. The rules of intervention are also not well understood. So we don't know really how to start changing these complex ecosystems. The common ways that things are being done now, and they are changing, is that you either introduce more of the agent that you're interested in. For example, in probiotics, if you look at the progression over time, we are going to higher and higher CFUs over time with the hope that if you introduce these probiotics, some of them will stick. But in a lot of cases, they don't, and they get just immediately washed away, and they don't have more than a temporary and transient effect. The same way with fecal transplant, that we don't really know what is happening. So we have to sort of trust that this new community that we are transplanting and introducing into a new environment will work well and won't cause too many side effects. And In some cases it does, and in some cases it doesn't. So then, given these complexities, the question is, what is it that we can do? And our approach has been to look into simpler examples as a way of learning the tricks of the job. So this is, I guess, maybe the roadmap. We start from finding a system that's relatively simple, not too complex, and it's tractable, so that we can do controlled and mechanistic studies in it. The second step would be to have a predictive model that captures the main properties of interest. And the hope of it is that this model, if it works, it will also is telling us, at least to some extent, that what we think about the community is actually what is happening within the community. So there aren't important processes that we're skipping. And because we don't want too much unnecessary complexity, the model should be as simple as possible and skip unnecessary details that are not important to our phenomena of interest. And then the third step is to use this combination of the tractable system and predictive modeling to design intervention strategies to change the state and figure out what are the limitations, what are the things that we can do, and what are the things that we potentially cannot do. And this intervention would be based on a constant iteration between the model and experiment. So we have to go through this process of figuring out if our model is good enough, does it capture the main process of processing in the experiments, and we'd go through validations to do that. So we started this process by first asking what would be the right system for us to use. And there are many options, several of them are good starting points, but we decided to use nasal microbiota because of its impact on respiratory health. It is known that microbes that are present in our nasal passage are important for preventing the colonization of opportunistic pathogens. As an example, the Staphylococcus aureus, if it's present in the nasal passage, it can cause bacteremia and other infections if the immune system stops doing its job properly. And that causes more than 100,000 invasive infections per year and around 20,000 deaths in the US each year. Staph aureus itself is present in the nasal passage. So roughly 30% of the human population carry Staph aureus in their nose, and around 2% of them even carry antibiotic-resistant staph in their nose. These are microbes that we have with them, and as long as our immunity is intact, they really are there just like any other commensal without causing many issues. But for people who have immune deficiencies or they are dealing with other health issues, staph can become pathogenic and cause really important infections that are hard to treat. So we asked if there are other microbes in that environment that could affect staph colonization and the risk of infection. Can we restructure the community? For example, in the 70% of the population that they don't have staph, what is it about them that suppresses them? And it is known now that there are several of these other commensals that suppress staph presence. So if we can potentially restructure the community and emphasize the presence of these suppressors of staph, then we can potentially get the result that we want and protect the patient from some of these infections. Nasal microbiota is also low in diversity, and that's an important aspect for us because we want to have a tractable system. This work by Skepa and colleagues in 2018 looked at the composition of nasal passage and looked at the species that were present in that environment. And just to orient you, the x-axis is the fraction of sequence reads in microbiome data, and the x-axis is the species numbers. And what you will notice is that there are a handful of species, essentially around 20 species that account for roughly 90% of the reads. And those are also the ones that are more prevalent, as noted by darker colors in the plot. So what it's telling us is that there are roughly 10% of these species that could be very diverse, but they are not very important. They are not everywhere. there are probably just transient species that come into that environment and go away without really staying in that environment. And there are these around 20 species that are essentially the core of the human microbiota that are often found in different microbiota and account for the majority of reads in these conditions. Importantly, around 19 out of these 20 are culturable in the lab. There is one that's still not culturable, but the majority of them are relatively easy to grow in the lab. Even if we look at the individual level, it's an even more striking picture that roughly 3 to 8 species account for the majority of reads for each individual. So that what's that telling us is that each individual essentially has somewhere between 3 to 8 nasal species that account for everything that account for the most important aspects of their function. And because we can easily grow them, we start these in vitro communities by taking nasal bacterial isolates, growing them in the lab, constructing small communities, but those small communities are not too far from what can happen in somebody's nose. And the idea is that we are capturing at least the majority of the diversity of the bacteria in that environment. In terms of modeling these communities, the most common and popular way of doing modeling is the Lotka-Volterra types of equations. They have a really long history in ecology. And the main selling point for Lotka-Volterra equations is their simplicity, that they look into the, sorry, I think there's a question about diversity. Sure, I'm happy to take it.
Yes, I just had a question. So you said that, in one of your examples, 20 of the taxa account for 90% of the reads. This is also true for the gut microbiome as well. There's sort of this there's kind of this long tail. So true that any other taxa, or does it also have this long tail? Right. So like, depending on what your thresholding is, you can say that there's between 100 and 300 ASVs in a single human fecal sample. But like I'm saying, that it's also this exact same phenomenon that, 20 taxa take up 90% of the reads. 99% of the reads are taken up by the top 50 taxa, but there's still tons and tons of other taxa. And also, when it comes to disease-causing bacteria or other things that are heavily studied, like all of Escherichia and Shigella account for less than 1% of the reads. C. diff is practically undetectable, even in cases where the person is sick and we have toxin detected. So, is it like the gut in that case, or here there truly are only like 20 species?
No, I would imagine there are. And that's a very good question and very important point, that it is. So there are other species that you can find in that environment. So if you take sample from an individual, you can find many environmental, or what we consider often as environmental microbes in that environment. I think in this case, what's unique is that these are easily culturable, which is not the same for the gut microbiota. So there is a little bit of barrier for constructing synthetic gut microbiota. But in the nasal situation, it's much easier because we know that these are the prevalent species and they are relatively easy to culture without having to work too hard. So I guess for us, what's important is that we can take these small committees and form them relatively easily in the lab. Does that answer your question?
Oh yeah, cool. I just thought it's something that I've always known about, the heavy tail of the reeds.
Right.
And I think sometimes, yeah, it's just fun to talk about because there's the nose probably is fundamentally different too, because you're just being bombarded by transient bugs. Compared to the fecal samples, right? It's a little bit harder to get all the way through there. But yeah, I was just curious.
Great, thanks. Just as a side note, I think the two aspects that probably contribute to lower diversity in the nose. One is the relative concentration of nutrients is low. So there is not a whole lot of nutrients in that environment, which is different from the mouth or the gut. And the other aspect is that there is a strong immune presence in the nose. So the immunity is actively suppressing things that come into contact with it in that environment, and that probably contributes to lower diversity in these environments. I think there's another question. Sure.
Yep.
Yeah, hello, can you hear me?
Yes.
Okay, I have one question. So your predictive model will be the interaction between the microbe that stays in the nasal the domain and the incoming, the intruder, and the immune system, our body immune system, right? So a three-way kind of interaction, our body immune system, and then the nasal bacteria is intervening, because directly that intruder, whatever the outside species, is interacting with that bacteria, and through that bacteria our immune system is cross-talking or something. Is that my understanding is, or is the multi-component more than three major component in the interaction?
So in our system, we even oversimplify it, and we have not included the effect of the immunity directly into our model. So for now, we are only focusing on the interspecies interactions, and I'll get into more details about that.
So,
I think it's fine. You can go. Yeah.
Okay. So the model that we started with is, of course, Lutko-Volterra type model, because of its long history and because of its simplicity, that only looks at how one species affects, or the abundance of one species affects the fitness of the other species, which is easy to quantify and easy to formulate in a model. And it's relatively modular. So if you look into these pairwise interactions, you can potentially build the network of interactions, all of that as well. And there is some empirical support for it, which suggests that maybe it's a starting point. So we started the Lutko-Volterra type model and asked whether this is applicable to microbial communities. And to do that, we took what we call a mechanistic model, which is essentially a series of species interacting through a series of metabolites, and each of the metabolites could positively or negatively affect the fitness of each of the species. And the idea was that maybe a typical microbial community looks like that. And we constructed a corresponding Lutko-Volterra type model by doing our best to capture how we can approximate this model using Lutko-Volterra type set of equations. And then we asked whether, when we simulate the dynamics, whether the results that we get from our model, which is, I guess, in this case, the ground truth, similar to the results that we get from the LV model. And the idea is that by doing this, we are avoiding the uncertainties about biological systems. So we are not relying on knowing exactly what happens in a biological system. Here, we do know what happens in the community to the finest detail. And we want to see what are the conditions under which these predictions match. And what we found from this exercise was that there are certain aspects that Lutko-Volterra type models are not good for representing microbial interactions. One of them is that depending on the interaction mechanism, they miss some of the aspects. And you can see that even in the simplest of cases, that you have two species, one is producing some metabolite that affects the other species, in this case positively. And we compared two cases. In one case, the metabolite was not being consumed by the recipient, and the other case, it was being consumed by the recipient. And when we compared these two cases, we saw that the results didn't match. So you could not use Lutko-Volterra type model to capture both of them. And the reason is almost trivial, that in one case, C is being distributed between different recipients. So the strength of interaction will depend on the abundance of the recipients, whereas in the other case, all of them are seeing the same thing the metabolite is not being consumed. So everybody is seeing the same regardless of how many of the recipients are present. So as you can imagine, these two types won't have the same type of equation to represent them. And we can see that Lotka-Volterra can't capture both of them at the same time. So you can come up with Lotka-Volterra-like equations for each of them, but not a single set of equations that would describe both. Another, again, almost trivial example is that in these situations that some metabolite is mediating the interactions, you can construct these simple cases that maybe for the interaction between 1 and 3, if you have another species that's consuming the same metabolite, its presence or absence will affect the interaction between 1 and 3. So if it's present, then there will be less of C1 present in the environment, and the effect of S1 interaction and S3 will be weaker. And that's the interaction that won't show up in pairwise characterization of interactions, which is commonly done for Lotka-Volterra type equations. So given these issues, we asked ourselves whether we should just abandon Lotka-Volterra type models and go to something else. But when we look back at the literature, there are some examples that, even though there are these examples that Lotka-Volterra seems inadequate, there are other examples that Lotka-Volterra seems to work just fine. So we asked ourselves, are there conditions under which Lotka-Volterra models would work well? And to test them, we go back to our nasal committees, which is sort of our bread and butter. These are committees that we can work with. They're easy to play with. And we ask those questions in this relatively simple model. So what we did was we characterized the interactions by looking at cell-free spent media of individual species and asking how other species were affected by it. So this is, we call them people's assay because it's the easiest type of experiment that you can do. It doesn't require fluorescent labeling or any particular manipulation. As long as you can culture the strains, you can run these assays. When you run these assays, what we get out of the CFSM assay is the growth rate and carrying capacities of species growing in supernatant of other species. And the idea is that each of these species are changing the environment by their activity. They are consuming some of the resources, spitting out other things into the environment. And when we expose other strains to that environment, then we can estimate how the activity of one cell type affects the other cell type. But importantly, what comes out of the Lotka-Volterra type equations is that if you run these experiments, the ratio of growth rate to carrying capacity in the CFSM is similar to the ratio of growth rate to carrying capacity in fresh media not affected by any other species. And what that means is that this is really independent of the species J. So regardless of what CFSM you're using, what the species it's coming from, the ratio should be constant. And there is a simple derivation that can show that. I won't spend too much time on the derivation itself, but the idea is that these are just coming from the Lotka-Volterra type model and estimating what is the growth rate and carrying capacity in CFSM of other species. And the net result is that this ratio is fixed. So we asked ourselves, if this is happening, then we should be able to use this as a way of testing whether Lotka-Volterra is a good set of equations for a particular system. So we can just run these assays, see if everything aligns on a line in terms of the ratio of R to K. And if they do, then at least things are consistent with Lotka-Volterra type equations. We know that if they are not consistent, then we probably can't use Lotka-Volterra for that set of species. And we did that with our nasal species. So what's being shown here is carrying capacity using maximum OD as the proxy for it, and the growth rate. So carrying capacity on the y-axis and the growth rate on the x-axis. And for each of these species, which is shown on top, we have looked at the CFSM coming from the other species that we often work with. And by just taking a quick look at it, you can see that this linear relationship seems to hold pretty well. There are some outliers. They could be experimental errors, or they could be something really biologically important that's happening in those cases. But for the majority of cases, the trend seems to hold. And that tells us that in this environment, which is about 10% THY, so we've reduced the concentration of a rich medium to bring the concentration of nutrients to levels similar to what has been observed and measured in the nasal passage. And we see that in that environment, things seem to align pretty well along that line. So R2K ratio seems to be constant regardless of where the CFSM is coming from. Then we asked what are the conditions that this will hold. And to begin with, we've asked whether it's just nutrient availability. So that's the easiest explanation, right? So you say that each of these species are taking up some of the nutrients that the other species need. And depending on how much they are taking away, then that means there will be some leftover, and R over K will be just somewhere along that axis. I think there is a question about the model equation, so I'll quickly go back. So this is the simplest equation for just looking at species 1 and 2, and including the logistic growth effect for species 1 and a single interaction term for accounting for species 2. Is that clear?
I suppose so.
We'll move forward then. So, and what we saw first was that the R2K relationship will hold if we just dilute the nutrients in the environment. So we started from 5% THY and just went through dilutions, that things to be aligned on that line. So it means that if the simple act of just reducing the amount of nutrients just creates that picture as well. Then we asked whether this holds everywhere, and we saw that in rich medium. So if we go above 10% THY, that relationship doesn't hold anymore. So something changes about the way species are interacting, that these more rich environments, even if we just do simple dilutions, they don't follow the same trend as what we expect for Lotkov-Volterra type equations to hold. And that tells us that probably rich media are not good for estimating the interactions or simulating the interactions between species either, because even if it's just a simple act of taking up glucose and reducing the amount of glucose in the environment, that's not enough in these rich environments because of the presence of other resources at abundance. That doesn't follow the Lotkov-Volterra type of trend anymore. The other thing that we tested was we looked at different individual mediators of interactions, things like amino acids, organic acids, and antibiotics. I'm just showing one of those examples in acetic acid here that is also shown, but the trend held in other cases as well, that if we use a single mediator, the trend doesn't seem to hold anymore. For some cases it does, and for other cases, either we see things are not sensitive or change in weird directions. And what that's telling us is that individual mediators probably don't create that pattern. So things have to be almost necessarily a little bit complex for the pattern to hold. And the sort of simple model that we have in mind about how these interactions are happening is that in low nutrient environments, there is a combination of effects that on average will keep us along this axis. So we call this axis like the habitat quality axis because it's telling us how the species grows in this environment. So originally in the fresh medium, it will be at some point, if the environment gets better, either by adding nutrients or providing some other benefit, then things will move up along the habitat quality. And if things get worse, if maybe a competitor removes some of the nutrients from the environment, then the habitat quality becomes less suitable.
I think there is a question from Daniel. Daniel, would you like to?
Sure.
I was just
Oh,
wondering
sorry.
if when
That's
you
a good
call it
question.
yeah. Okay.
So the rich, the way we are talking about here is the concentration of the nutrients, not the number of resources. Sorry, I should have been more explicit about it. Thanks for the clarification. So, and what we think is happening is that we have these different effects from different mediators in the environment. So there is changes in the organic acid concentrations, changes in amino acid concentrations, and each of them are pulling the system in some of these directions. But on average, it's still the nutrient availability which ends up being the majority of the effect. So all the other effects on average, in these relatively complex environments, and complex in the sense that there are many different mediators present in the environment, then on average, we will follow this trend along the habitat quality that either negatively or positively affects the species. And in some sense, this complexity of the environment in terms of the number of mediators, which will be really hard to track individually, leads to this relatively simple model of just looking at one axis and determining how one species affects the other species. Just to confirm our results, we looked at co-culture experiments, and I'll go quickly through these. So these are, we have one fluorescent Staph aureus strain, and we put it together with another non-aureus Staphylococcus species from the nasal passage. And we use the fluorescence of the staph to determine the abundance of staph in the co-culture, and then use the overall OD and subtraction of the known amount of staph to estimate how much of the other species, the non-fluorescent one, is present in the environment. But in the end, what we see is that the models that we get from CFSM data, which is just very simply estimating what the interaction coefficient is, provides a decent approximation about what the dynamics would be. And there are three different plots in this case. The dotted line is coming from the CFSM data. The solid line is the actual experimental data. And then the dashed line is our best effort to fit a local ultra type into the model. So if we fit a model into it, it will be better than the CFSM model. But overall, when we compare the interaction coefficients between different cases, things more or less match. And independent of the initial ratio, they more or less match. And what that tells us is that in these simple cases, in, again, low nutrient rich environments, it seems like the CFSM characterization is good enough to give us at least a basic idea of how the interactions are and what the dynamics would be. We did the same thing with rich medium, and as we expected, the results didn't match. So we can still find a fit in by just directly fitting a local ultra type in the model, but that fit changes quite a bit when we just change the ratio of species. So things are not consistent, which means probably we can't find the local ultra type model that's good enough to give us the dynamics. And this is inconsistent with what we expected already, because we know that the linear trend between growth rate and carrying capacity doesn't hold in these environments. We took one more step and asked ourselves whether, when we look at combinations of species, whether we can go beyond just two species. And because we don't have enough fluorescent strains, we did this shortcut that we took the CFSM from different species, combined them together, and asked whether the combination of CFSM media was equivalent. The response in the combination was the average of the response in individuals. So that tells us that if you have two species, each of them are affecting the environment in some way. If you combine them, if both of them are present at the same time, whether the results will, the local ultra type will still hold or not. And for the majority of cases that we tested, it did. So we didn't see anything too jarring about the way the combination of CFSMs worked. But there's still more work to be done to confirm things, even with three species, or even go beyond 2 and 3 species and ask how far can we push local ultra type model before things fall apart. And then we'll get to the intervention part. So to investigate intervention, we chose invasion as our building block. So you can do intervention in many different ways, and we decided to look at invasion as the way that we examine how well we can intervene. The reason we did that was that the invasion itself is a relatively well-defined process, so you can have a clear readout whether the invasion was successful or not. There is a long history of invasion ecology, which gives us a lot of conceptual framework about what to look and what to avoid. And it's also practically relevant because for situations like introducing a new probiotic, we want to make sure that invasion is successful without disrupting the resident community. So that would be relevant in cases that we are considering here. And just as a side note, the two concepts that we go to are colonization resistance, which means how the community can protect itself from invaders, and engraftment, which is the act of introducing a new species into an existing community. So these are the typical things that we think about when we are talking about microbiota and intervention. And to begin with, we started this theoretical investigation of asking how interactions between the species could contribute to the success or failure of invasion. And to do that, we built a theoretical assay and a model and a corresponding model in which we made certain decisions about the assumptions. One of those assumptions is that we reduce the effect of resource competition by simulating the situation that cells are going through cycles of growth and dilution. So the shared nutrients in the environment are constantly being supplied from the environment. It's closer to a continuous culture situation. And in those cases, we don't expect too much effect from just background resources that are provided by the environment. And the other thing that we did was that we looked at the interactions that, again, were mediated through metabolites. And the idea was that this would give us more flexibility to examine what happens if something is not being consumed or is not being produced, compared to the basic blood culture type model, which has only one interaction coefficient to play with. And then we introduced the invader into an existing resident community, which is already stable, and asked what happens to it. And we imagine there are 4 possible outcomes possible. Augmentation, which means both the invader and previous members are maintained in the environment. Displacement, which means the invader exists, but some of the existing species go extinct. Resistance, which means the invasion fails, or disruption, which the invader fails, but some of the previous members also go away in that process, go extinct in that process. So these are just 4 possible outcomes that you can see. And then we asked what happens under different situations to the frequency of these outcomes. And the first thing that we are looking at here is the effect of propagule size of the invader. So what happens if we add more and more of the invader into the community? And what we see that in this situation, which again, the interactions between species is overemphasized in our model. In this situation, we see that the invader frequency is relatively unimportant unless we introduce a lot of the invader into the system. And even in that case, what happens is that instead of resistance, we see more disruption and displacement. And this is sort of a warning sign for trying to introduce the probiotic by just adding more and more of the probiotic. Again, these results are not universal. These are just simulations under certain conditions. But in these conditions, we see that that process is probably not helpful. And it can only lead to disruption and displacement rather than just augmentation, which is the ideal outcome. We also looked at the effect of the interactions either within the community or between the community and the invader. And the plot on the left-hand side is showing the effect of changing the inhibition on the invader. So on the baseline model, we assume that the existing resident community inhibits the invader, and we here are modulating that effect and allowing them to be more helpful to the invader. And as you would expect, moving to the right, you will see more and more examples, less and less resistance, that because there is some support for the invader, it becomes more and more likely for the invader to establish. We also see more examples of augmentation in those cases. Again, as you would expect, that if the community is supportive of the invader, there will be more chances of invader just assimilating into the community. The other factor that we looked at was what happens if we change the interactions within the resident community. And what we see is that in communities that have more facilitation among them, so these are more cooperative communities, they have stronger resistance and lower chance of displacement. And if we push things to the edge, so make more and more competitive communities, then those are situations that we'll get more examples of displacement. And then we sort of reorganized our thoughts about asking, knowing what we know now, what is the impact of modifying the interactions on the outcome of invasion? And again, we're using this simulation platform to make predictions. But to simplify things, instead of looking at the details of the resident community and invader, we assumed the resident community as a unit and invader as a unit, and categorically changed the interactions between them. So instead of assuming what happens to the details of these interactions, we are just asking what happens if the invader doesn't produce any of the metabolites, or if the inhibition from the community as a whole to the invader is reduced. And
sorry, we have a question regarding the value. So I don't know actually the name of who's asking, but he's asking about the threshold value necessary for invasion.
So that depends on the details of how you set up the experiment, or I guess in this case, the simulations. I can't give you an exact number, like what would be the threshold for it to work. But I can tell you that there are these trends that if you add more invaders, then you're more likely to get, for example, displacement and disruption. Sorry, if this is not the parameters that are important are the frequency of interaction types and the basal growth rates of the community members and invaders, and all of that right now in this practice, in this exercise, it's all hypothetical. So we just put numbers that are comparable to what we see experimentally, but they are not reflecting anything, any exact biological system. So what we did was we made these changes. So dropped, for example, the production links and asked what happens to the outcomes. And I'll skip through most of the details, but there are two trends that are very easy to see. One is that when we allow the community to produce some of these metabolites that are beneficial within the community, so in some sense the invader is providing a benefit to the community, then we get more chances of assimilation into the community or augmentation. And the other easy trend to see is that when we drop the inhibition of the invader by the resident community, then there is a huge shift and gives us less resistance and more examples of displacement in those cases. But that basically what's telling us is that we can manipulate the interactions and get some of these results that we could potentially be interested in. But this is all, again, theoretical investigations. So we wanted to go back to the experiments and see how we can actually implement these ideas. One of the graduate students in the lab, Vaishnavi, is doing this job. This is essentially the roadmap of what she's following. She is building stable communities of nasal microbiota in the lab. So these are our baseline resident communities that we can use to investigate different invasion outcomes. Then we introduce an invader and probe the outcome. She's using a fluorescent strain as an invader, which makes it easier to detect what happens to the invader. And then in parallel, we're constructing models for these slightly more complicated situations that we have the resident community and the invader all present at the same time to see whether these models are predictive of invasion outcomes, which is a slightly easier question to answer compared to predicting the actual dynamics of the system. And then we want to see if we can use the model to predict what sort of intervention is more effective, and we'll validate those experimentally. Just to give you some of our progress, she has already established several stable communities. So these are communities of 2 or 3 species. And essentially what we do is that we go through rounds of growth and dilution, and often after about 4 rounds of dilution, regardless of what the initial ratio was, the community seems to stabilize at some level. And that's the stable communities that we can use then sort of with maybe a slight leap and sort of telling ourselves that these could be somebody's nasal microbiota. So then we want to see how these communities respond to invasion. And for that, essentially, we build these stable communities first. We introduce the invader. We watch what happens to the invader after maybe 1 or 2 cycles of growth. And then that would be our final community that we can use to make calls about whether the invasion was successful or not. Just to give you some, these are very recent results, so they are still not very refined, but I wanted to share some of the things that Vaishnavi has found. So here what she's doing is she's monitoring the GFP, which is coming from the invader, introducing the invader at different ratios initially and asking how the ratio of GFP to overall OD changes over time. So when we normalize it, essentially this is telling us to some extent what's happening to the invader. If this goes up, it means there will be more of the invader. And if it goes down, there are fewer invaders present relative to the rest of the community. One thing that she noticed and we are trying to correct for is that scattering from non-fluorescent cells can't be ignored. So if you have some fluorescent cells and a lot of non-fluorescent cells, then the amount of fluorescence that you will detect will be lower than expected. But she has done some basic calibration and we are in the process of doing that correction so that we are sure that the GFP that we are seeing here is the actual GFP rather than just the detected GFP affected by scattering. This is all work currently going on in lab, but I'm very hopeful that in near future we'll have results from these invasion assays. And that's pretty much it what I wanted to cover. And just to quickly summarize, we think that these in vitro communities of sorry, just quickly going back. So the OD is the OD of the cells in the environment, which includes the OD from the fluorescent cells as well as the OD of non-fluorescent cells. And what we see is that if you have more and more non-fluorescent cells present in the environment, the fluorescence from GFP cells will be lower and lower because some of it will be lost to the scattering. Hope that makes sense. Right, so we think that nasal microbiota would be a good and tractable system to do these kinds of experiments, which gives us essentially the basic idea of what are the things that we can do, what are the limitations of our tools, such as the models that we are using and the intervention plans that we have when we are moving towards more realistic communities. And the hope is that these will be the building blocks of starting to look into really more complex communities, which include some of the aspects that have been mentioned. For example, the presence of low abundance species or the presence of some other factors such as the immunity in the environment. Thanks, and I'll be happy to answer any remaining questions.
So thank you, Babak, for the presentation. It was really clear and interesting. So now before question, actually, I will stop recording.